So in this chapter, we're going to be looking at cellular respiration. So this is going to be how we get energy in animal cells. Plant cells also do have to do some cellular respiration, but when it comes to plant cells, we focus on photosynthesis since that is their unique process. So in cellular respiration, we're going to be looking mostly at the catabolic pathways that are going to break things down and yield energy by oxidizing organic fuels. So what you'll do is you'll have the potential energy be released in the exergonic reactions. Fermentation involves just a partial degradation of the sugars. This occurs without the use of oxygen. Aerobic respiration means that oxygen is going to be consumed as one of the reactants along with the organic fuel. Cellular respiration technically is going to include both aerobic and anaerobic processes. It's sometimes called aerobic metabolism, or cellular respiration has the aerobic metabolism as a component for it. The term respiration originated as a synonym for aerobic respiration because of the relationship to breathing oxygen. So it often is going to be referred to as aerobic respiration. So when we look at the general reaction that occurs here, you start out with an organic compound that's going to be reacted in the presence of oxygen to yield carbon dioxide, water, and energy. The one we specifically look at here is glucose, the C6H12O6, will react with 6 oxygens to give you 6 carbon dioxide, 6 water, and energy in the form of ATP and heat. The delta G for this reaction is negative 686 kilocalories per mole, so it is a spontaneous reaction. The catabolic pathways themselves are not going to directly perform the cellular work. Instead, they're going to be linked by ATP as a chemical drive shaft. It's similar to the idea that you go to work, you get paid with a paycheck. You use that paycheck to pay your other bills. The cell is going to break down glucose. It's going to store its energy in the form of ATP, and then it will use ATP to fuel other processes in the cell. So these are going to involve some redox reactions, sometimes called oxidation and reduction. With these reactions, you're going to have the transfer of electrons. So the principle of redox is that you transfer one or more electrons. Redox is also called oxidation reduction in chemistry. Oxidation is the loss of electrons from one substance to another, and reduction is the gain or addition of electrons from one substance to another. These reactions generally occur together because you're going to need a source of electrons for somebody to gain, and if you get rid of electrons, they have to go somewhere. Your reducing agent will be the electron donor, and the oxidizing agent is the electron acceptor. The mnemonic that chemistry typically uses to remember this is oil rig. Oxidation is loss, reduction is gain of electrons. So a reaction does require both a donor and acceptor, so these reactions are almost always going to occur together. Not all redox reactions will have the complete transfer of electrons. Sometimes there is a degree of sharing. So this image here is not real great for seeing who is oxidized and reduced. It's a little bit better in the picture below. When we oxidize organic fuels during cellular respiration, we are going to have part of the reactants oxidized and part are going to become reduced. In the process of doing that, the electrons lose some potential energy along the way, and that energy is released. The state of the electron changes as the hydrogen with its electron is transferred. If we look here, you've got methane, the CH4, that's going to become oxidized to become carbon dioxide. Here, the oxygen becomes reduced to become water. Down here, you can see where the electrons actually move during this reaction. In here, I'm not overly concerned about you being able to follow where the electrons move necessarily. I do want you to remember, though, that it is redox reactions involved in this. So stepwise, the energy harvested via NAD+ in the electron transport chain is going to be an important part of this. Instead of breaking things down all at once, we break it down in steps, and this is going to allow the energy to be harvested and provided in smaller amounts to be able to do work. In key steps, you're going to have electrons removed from the glucose, and they'll travel with a proton as hydrogen. But they're not going to be transferred directly to oxygen. Instead, they're going to pass through this carrier that is going to be known as NAD+, nicotinamide adenine dinucleotide. The enzyme dehydrogenase removes the hydrogen pair from the substrate and oxidizes it. NAD+ is a very versatile electron acceptor in respiration. We will use it at several steps. The reason we use it is that the electron loses very little potential energy when it's transferred from glucose to NAD+. So it's an efficient way of being able to transfer it. It's not an expensive way of being able to transfer it. When hydrogen is going to combine with the oxygen to make water here, it would be an explosive release of energy. So we do want to do that stepwise so that we can harvest the energy in a usable amount. As it gets passed down the line, you're going to have each downhill carrier be a little bit more electronegative than the uphill carrier, and it oxidizes the uphill neighbors. So this is going to keep the electrons moving down the chain, just like gravity pulls an object down the hill. This is showing NAD+ here, when you have the hydrogen ions and the electrons here, and when it becomes NADH. So you can compare the two forms of it. Where you can see the difference here is this little hydrogen versus only one of them here. In cellular respiration, we have several major stages that we're going to look at, and we're going to break each stage down into details. The first stage will all be in green. It's glycolysis. This occurs in the cytosol, and the overall reaction is to split glucose in half to make 2-pyruvate. The citric acid cycle is going to occur in the mitochondria. So we're going to have a step here where we convert pyruvate to acetyl-CoA in getting it into the mitochondria. This would occur in the cytosol of prokaryotes, since they don't have mitochondria. Your complete breakdown of glucose will happen by oxidizing the derivative of pyruvate to carbon dioxide. So this acetyl-CoA is going to come in, and you're going to have everything get oxidized to make carbon dioxide. In some steps, you'll have the electrons transferred onto NAD+ to make NADH. The final step will be in purple. This involves your electron transport chain. This is where you'll do the oxidative phosphorylation. The electrons are going to get passed along a chain of protein carriers, and at the end, they will be combined with oxygen and hydrogen to form water. And the energy that gets released is going to be enough to make ATP through oxidative phosphorylation. This image here is showing that when you have that combination of the hydrogen and the oxygen here, it's an explosive release of heat and light, versus doing it stepwise here in the electron transport chain in cellular respiration. You get a controlled release of energy for ATP synthesis. When we talk about phosphorylation, there's two different ways it happens. Substrate level phosphorylation is going to have a small amount of ATP be formed in glycolysis. It will involve an enzyme transferring a phosphate group from an organic substrate onto the ADP. With oxidative phosphorylation, you use an inorganic phosphate. So it's still phosphorylation either way. Substrate level, it comes from an organic substrate. Oxidative phosphorylation, it comes from an inorganic phosphate. For each glucose that gets oxidized all the way to carbon dioxide and water by respiration, it will produce about 38 ATP, or a net gain of 36 ATP with 73 kilocalories per mole of free energy formed. The number that you want to remember here is this net gain of 36 ATP. That's the number you're more likely to run into over and over again. What we're going to do now is look at the steps of glycolysis. This is an overview of glycolysis here. There are two phases to it. You've got an energy investment phase where you're going to have glucose, and you're going to put the energy from 2 ATP into the reactions. Then the energy payoff phase, where you're going to get the energy from 4 ATP back out of the reactions. You're also going to transfer some electrons, and gain 2 NADHs. And your glucose in this process has been split into 2 pyruvates and 2 waters. When we look at the net reaction here, the glucose becomes 2 pyruvate and 2 waters. You have 4 ATP formed. You had 2 in your initial investment during the energy investment phase, so that's going to leave us with a net gain of 2 ATP. With the NADH, you're going to have 2 of those pick up 4 electrons to make 2 NADHs plus 2 hydrogen ions. So if we look at the details of this here, they are written out in your notes, but they're also easier to read on this diagram of the chemical reactions here. We start out with glucose. Glucose has 6 carbons. ATP is going to be taken. You're going to take the 3rd phosphate off of ATP, which will leave it in the form of ADP, and it will transfer on to carbon number 6, which is why we call it glucose 6-phosphate. The number 6 just means that the carbon is on, or that the phosphate's on carbon number 6. Down below here it lists the particular enzymes that are involved in each step. Each step will have a set of enzymes that regulates it. This glucose 6-phosphate will rearrange itself here to make fructose 6-phosphate. We still need to put a little bit more energy into this reaction, so a second ATP is going to be invested, and it's going to put its phosphate here on carbon number 1. So now we have fructose 1,6-bisphosphate. The bis just means 2 of them. They're on carbon 1 and carbon 6. At this point, there's enough energy in here that the bonds will break and split it in half. On one half, you'll have dihydroxyacetone phosphate, the other one glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate is what moves on to the next step. The dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are interchangeable with each other. They can convert back and forth. Since the glyceraldehyde 3-phosphate is going to be the one that always moves to the next step, equilibrium is always going to drive the dihydroxyacetone phosphate to all convert to glyceraldehyde 3-phosphate. So then we'll put in some inorganic phosphate here, and we are going to move some electrons, so we'll get 2 NADHs. From this step on, you have 2 of each of these molecules because it was split in half. So now we'll have 1,3-bisphosphoglycerate. At this point, we're ready to pull some energy off. We're going to pull off one of the phosphates and transfer it onto ADP to make ATP again. Remember, there's 2 of them because there are 2 molecules of this. So then we'll have 3-phosphoglycerate. It will rearrange to be 2-phosphoglycerate. It will rearrange to become phosphoenolpyruvate, and we will finally pull off this last phosphate here and put it on ADP to have your net gain of 2 ATP, and we're left with pyruvate. So just to come back and see where you came from, this is glycolysis that we just talked about, where glucose is converted to pyruvate. This is your summary of what you got here. Glucose made 2 pyruvates and 2 waters, a net gain of 2 ATP, and 2 NADHs released from the reaction. So next we're going to look at how do we get this pyruvate into the mitochondria. So this of course is in a eukaryote, a prokaryote. This will all occur in the cytosol. Here you've got pyruvate and you've got a transport protein that's going to move this into the mitochondria. As it goes in, you're going to pull off the carbon and 2 oxygens as carbon dioxide. You're also going to move some electrons, so you'll have NAD plus be converted into NADH here. What you end up with is this 2-carbon molecule that will join with coenzyme A to make acetyl-CoA. Acetyl-CoA will be ready to move on to the next step, the citric acid cycle or the Krebs cycle. So the citric acid cycle does have multiple names. It's also called the Krebs cycle or the TCA cycle or tricarboxylic acid cycle. They're all correct. This book uses the citric acid cycle. Your acetyl-CoA is going to bring 2 carbons in. They're going to join with these 4 carbons of oxaloacetate. When you take the 4 carbons plus the 2 carbons, you're going to make a 6-carbon citrate. It will rearrange itself a little bit to become isocitrate. And then it's going to pull off a carbon that will combine with 2 oxygens, and in the process remove some electrons that will become NADH and more carbon dioxide. The carbon dioxide here is exhaled from cellular respiration in each of these steps. So now we're down to the 5-carbon alpha-ketoglutarate. We're going to remove another carbon here, combine it with oxygen. It will be released as carbon dioxide, and moving some more electrons, NAD plus becomes NADH. Now we are down to the 4-carbon succinyl-CoA. You can almost think of these NADHs as being like an alternate energy currency. If ATP is like your dollars, that's your preferred currency. Having these NADHs is another currency storing energy that would be like having some euros. The succinyl-CoA will be converted into succinate. In the process, you're going to have an inorganic phosphate here that's going to come on and turn GDP into GTP. That GTP will then transfer the phosphate onto ADP to make ATP. So the GTP is like having another energy currency again, but this one just converts right away to ATP. The succinate will rearrange to make fumarate. In the process, moving some more electrons into yet another way of transferring them, FAD, that will become FADH2. The fumarate will rearrange to malate, the malate back to oxaloacetate from the beginning, and also releasing more electrons on the NAD plus to become NADH. When you start with 1 glucose, you're going to get 2-pyruvate. 2-pyruvate will end up producing 2 acetyl-CoAs, which is 2 turns of this cycle. So for each glucose, you will have 2 turns of the citric acid cycle, so twice as many of each of these things released that you see coming off of the cycle. So where we are in this big picture, if we come back up here, we've looked at glycolysis, how you take the pyruvate and convert it into acetyl-CoA, and the acetyl-CoA is going to come into the citric acid cycle. We've seen where we produce the ATP through substrate level phosphorylation and glycolysis. We've seen where we produce the NADH. Here we've seen where we produce the ATP, whether it be directly as ATP or as GTP, and where we're going to have the electrons moved and produce NADH and FADH2. At this point, we're going to look at the electron transport chain where we're going to do oxidative phosphorylation and chemiosmosis to get the last of the ATP. So during oxidative phosphorylation, chemiosmosis is going to couple the electron transport to ATP synthesis. In the electron transport chain, you've got a collection of molecules that are embedded in the inner membrane of the eukaryotic mitochondria. The chain is going to have multi-protein complexes. So we have complexes 1 through 4 here. The carriers will alternate between reduced and oxidized states as the electrons move down the chain. So here you're going to have NADH that's going to drop off its electrons and become NAD+. FADH2 is going to cut in line a little bit here. It's going to drop them off on the second protein complex, and then the electrons will be passed down the chain all the way to the end. So in this picture, it's going to illustrate things a little bit better, I think. Here the NADH drops off its electrons. You can see the electrons are going to be passed down the line, FADH passing its electrons down. When you pass the electrons through, each time you go through one of these protein complexes, it's going to pump a hydrogen ion onto the other side of the membrane. So NADH ends up pumping 3 hydrogen ions onto the other side of the membrane. FADH2, on the other hand, because it cuts in line a little bit, is only going to pump 2 hydrogen ions across the membrane. This separation of the hydrogen ions and the electrons is going to be what creates chemiosmosis. You're going to have the energy stored in the form of this hydrogen gradient across the membrane. ATP synthase is an enzyme at the very end of the chain that is going to make ATP from ADP. The proton motive force is going to be when the electron carriers deposit the electrons in the inner membrane space to create the hydrogen gradient. This gradient has the capacity to do work. This is an enlarged picture of the ATP synthase enzyme. So you have this rotor and stator. What will happen is the hydrogen ions are going to attach to the rotor. It's going to spin around before it drops that hydrogen ion down another little chute. And when it spins that rotor around, it's attached to an internal rod that spins the catalytic knob, and that will cause the ADP to bind to the inorganic phosphate and become ATP. When we look at the conversion of this, you're going to have each NADH make 3 ATP. Each FADH2 is going to make 2 ATP. So from the citric acid cycle, you're also going to get 2 ATP by substrate level phosphorylation that were first converted to GTP and then to ATP. So what enters into the electron transport chain are 6 NADHs and 2 FADH2s. Your citric acid cycle will give you 2 ATP by substrate level phosphorylation. When we look at the totals of this, your 6 NADHs or 2 FADH2s will give you 32 or 34 ATP, depending on which shuttle transports the electrons from NADH in the cytosol. The total maximum glucose is about 36 ATP. So it's 38 ATP if you're not taking out those initial 2 you invested in glycolysis. If you take out the initial 2 you invested in glycolysis, the net gain is 36 ATP. These numbers are technically not real exact because you can have different carriers be used, but the numbers that you do want to focus on are the 6 NADHs, the 2 FADH2s, each NADH giving you 3 ATP, each FADH2 giving you 2 ATP, and your net gain being a total of 36 ATP. This here summarizes where you get everything. So glycolysis, glucose to 2-pyruvate, 2 NADHs, 2 ATP. We convert the pyruvate into acetyl-CoA. Excuse me. Acetyl-CoA in that process is going to give you 2 NADHs. These 2 NADHs are going to transfer to the end in the electron transport chain. During the citric acid cycle, you're going to get the 2 ATP via 2 GTP, 6 NADHs, and 2 FADH2s. When you go through and you add everything up, you will be able to figure out where you get the net gain of 36 ATP. The complete oxidation of glucose under standard conditions is negative 686 kilocalories per mole. Phosphorylation of ADP to ATP is 7.3 kilocalories per mole. So if we look at the efficiency of respiration, you have 7.3 kilocalories per mole times 38 moles. We're using the overall number. We're not taking out the initial investment. And you take that number here. So if you were to do that on a calculator, .3 times 38 is going to give you about 277. When you divide that by the 686 kilocalories per mole, what it comes out to be is about 40% efficiency. When you initially hear that, you may not think that that's too impressive. However, 40% efficiency is pretty good. And it's actually probably higher because delta G is lower under cellular conditions. The rest of it is lost as heat. The heat isn't really unimportant here. It is important because we use it to maintain body temperature. It can also dissipate as sweat, so it does play an important role in our lives. 40% doesn't sound great, but the most efficient automobile only converts about 25% of its energy, so better than anything we've been able to create. When we look at anaerobic respiration, there are certain anaerobic prokaryotes that have electron transport chains that don't use oxygen as the final electron acceptor. In our electron transport chain, oxygen is the final electron acceptor. Here, where we are dumping off these electrons, is here on the oxygen. An example would be the sulfate-reducing bacteria. They're going to produce H2S here instead of water, so they will produce a sulfur product instead. Fermentation is going to be the harvesting of chemical energy without the use of oxygen or an electron transport chain. In order to do this, you need a sufficient supply of NAD+ to accept electrons during glycolysis. There are two main types of fermentation we look at here. There are many other types of fermentation that bacteria can do. Fermentation consists of glycolysis plus a reaction to regenerate the NAD+ by the transfer of electrons from NADH to pyruvate or its derivatives. If we look at alcohol fermentation here, we start out with glucose. It goes through glycolysis, and in the process you generate these two NADHs. We're going to take the 2-pyruvate, and carbon dioxide is going to be pulled off, and you will produce 2-acetaldehyde. The acetaldehyde will be converted into ethanol. In the process of converting it into ethanol, you're going to take 2 NADHs and convert them back to 2 NAD+. So the importance of this is not necessarily the ethanol. We do use it for commercial products. However, the reason that you make the ethanol is because you need to have enough NAD+ to sustain glycolysis. This is where you're getting the energy in glycolysis, not from actually making the ethanol. The ethanol just allows you to have the NAD+ to sustain glycolysis. With this, you do have carbon dioxide produced as a byproduct. With lactic acid fermentation, you're going to have the glucose be converted into pyruvate through glycolysis, and then the pyruvate will be directly converted into lactate. When you take the pyruvate and make it into lactate, you're going to take the 2 NADHs and turn them into NAD+. So again, here you can have the NAD pluses be available to sustain glycolysis. When we compare aerobic respiration and fermentation, both are going to use glycolysis to oxidize glucose and have a net gain of ATP. NAD+ is going to be the oxidizing agent that accepts the electrons from food during glycolysis. So in the comparison here, when you get to pyruvate, it will make its decision based on whether or not oxygen is present. Oxygen is present, it's going to choose to go with aerobic cellular respiration. It's a lot more efficient. If oxygen is not present, it will go with fermentation and make your ethanol, lactate, or other products. The difference in them is how they get NADH back to NAD+ to sustain glycolysis. In fermentation, the final electron acceptor is an organic molecule. So pyruvate for lactic acid, acetaldehyde for alcohol. In aerobic respiration, the final electron acceptor is oxygen, and it's going to have a lot more ATP come from it. Aerobic respiration is going to yield 19 times more ATP than fermentation. So it's much more efficient. When we look at the types of organisms out there, some are going to be able to use oxygen, some are not. The obligate anaerobes are going to be organisms that carry out only fermentation or anaerobic respiration. They can't survive in oxygen. Oxygen is toxic to them, so they have to use fermentation as a way of getting energy. Obligate aerobes are only going to carry out aerobic respiration. They don't do any fermentation. Vertebrate brain cells are actually going to be in this category. If you look at how long humans can survive without oxygen, it's not very long because of the damage to the brain cells. Facultative anaerobes, these make enough ATP to survive using either fermentation or respiration. Your muscles are going to be this way. They're going to do aerobic respiration when they can, and when they can't sustain aerobic respiration or they need more energy than what's available, then they will also start to do some anaerobic respiration. The evolutionary significance of glycolysis. Ancient prokaryotes probably used glycolysis to make ATP before oxygen was present in the atmosphere. The cyanobacteria produced oxygen gas as a byproduct of photosynthesis, and allowing this oxygen to start to build up in the atmosphere would allow the development of aerobic respiration. So our catabolic pathways have a lot of versatility. What we've looked at is the pathway of glucose. Glycolysis will actually accept a wide range of carbohydrates. Most of them get hydrolyzed to glucose or other intermediates to join the cycle. So if you start out as a more complex carbohydrate, it will be broken down into its smaller pieces to either be glucose or other simple sugars, and then it will join in near the top of the cycle. When we look at it here, you've got glycolysis, other sugars will join in near glucose. Proteins, when these are consumed, they have to be digested and broken down into the amino acids. These amino acids can actually be used to build new proteins, and they can detour off the pathway. The ones that are left are going to have to undergo deamination. Here you're going to pull off the nitrogen waste. In humans, this happens in the liver, and it will be removed as urea through the kidneys in your urine. The remaining amino acids are going to either join in at pyruvate, at acetyl-CoA, or somewhere in the citric acid cycle. So the ones that can come in at pyruvate are sometimes referred to as glucogenic because they do have the potential to be converted back into glucose. The ones that come in at acetyl-CoA or later are sometimes referred to as ketogenic because they will produce a ketone as a byproduct, which isn't shown on this picture. The triglycerides, these are going to be the form your fats are going to come in. They will have a glycerol molecule with 3 fatty acids attached. The glycerol molecule will be pulled off and it comes in at glyceraldehyde 3-phosphate. So it has the option of being converted back into glucose or into pyruvate. The fatty acids are going to come down here at acetyl-CoA. Once you get to acetyl-CoA, this is an irreversible step in the process, and you are converted or committed to no longer being converted back into glucose. So your fatty acids are going to undergo beta oxidation, where 2 carbon fragments at a time are going to break off and enter in at acetyl-CoA to go through the citric acid cycle. Biosynthesis, how we're going to build things. This process also provides us with a lot of building blocks. The cells can use some of the carbon skeletons to make their own molecules. Your amino acids from the proteins can be converted into your body's own proteins that it needs. Your intermediates of the citric acid cycle and glycolysis can be diverted into other anabolic pathways when needed. When excess food is taken in, whether it's carbohydrates, proteins, or fats, they're all converted to fat stores. So excesses of anything ends up being fats. So regulation of this is through feedback. The most common way is feedback inhibition, where you're going to have an end product of an anabolic pathway inhibit earlier enzymes to shut things down. So I'm not too concerned about you knowing these specific examples here, but these are just a couple of examples where you can have ATP levels at strategic points. Step 3, where you can inhibit phosphofructokinase. That can be sensitive to citrate in the citric acid cycle and help to synchronize the rates of glycolysis in the citric acid cycle so that you're not having one cycle go significantly faster than the others. So this is a tough section. What I would recommend doing for your studying is going back, studying your pieces of glycolysis, and then walking away and just let the information settle in. Study your pieces of pyruvate oxidation in the citric acid cycle and let that sink in. Then study the oxidative phosphorylation in the electron transport chain. Break it into smaller sections and try and just digest it in smaller bites. What you want to really focus on is the key things. What does the cycle start with? What does it end with? Where are you making the ATP? Where are you moving the electrons with NADH and FADH2 throughout the process?